Hi everyone. Now we are going to discuss a topic called screening and enumeration of microorganisms. As a part of daily routine, the laboratory microbiologist often has to determine the number of bacteria in a given sample as well as having to compare the amount of bacterial growth under various conditions. Then what is meant by screening and enumeration? The screening is a type of procedure used to isolate, detect, and separate the microorganism of our interest from a mixed population by using highly selective procedures is called screening. So we can get the microorganisms from the different sources like from the environment, air, water, soil like that, and from the food samples like meat, milk, cheese, etc. The enumeration of microorganisms is especially important in food microbiology as well as in dairy as well and water microbiology too. Knowing the bacterial or the microbial count in the drinking water or fresh milk, buttermilk, yogurt can be useful in many aspects of our food industry or food what we call it as food microbiology or industrial microbiology. So this food is going to serve as an excellent substrate for the growth of different kinds of microorganisms that we know. And the microorganisms enter into the food and grow as a contaminants or intended additions. The growth of these microorganisms in food may spoil food quality, and consumption of such food creates the hazard health effects in humans as well as animals. That means they are being led to the foodborne illness. So that we have discussed the food quality, foodborne illness, all these things in the previous videos. You can go through it. So the microbes and their enzymes are the major causative agents of our spoilage of our food. The growth and the resultant biochemical activity of microorganisms is going to be supported by the different factors such as intrinsic factors as well as the extrinsic factors which exist in the food and the environment. Coming to the types of spoilage microorganisms, based on the biochemical characteristics and the nutritional requirements, spoilage microorganisms can be categorized into these types. Number 1, psychrotrophic, thermoduric, lipolytic, proteolytic, pectinolytic, halophiles, osmolophiles, then acid producers. So these are the few types of spoilage microorganisms that we are having in the food especially, and this classification was mainly based on the biochemical characteristics and the nutritional requirements. First one, psychrotrophic count. That means how you are going to enumerate the psychrotrophic organisms or psychrotrophs. So the organisms growing at low temperatures, that means at the refrigerator conditions, are going to be called as psychrotrophic. The best examples of these psychrotrophic organisms are mainly belonging to the gram-negative, which are thin rods, that is bacilli, like Pseudomonas, Achromobacter, Flavobacterium, and alkaligens. And these organisms is going to cause the taint production, that is off taste and off odor is going to be called as a taint production due to their proteolytic as well as the lipolytic in the food. In food stored under refrigerated conditions for longer periods, these are going to cause this kind of a spoilage called as taint production. The excessive growth of these psychrotrophic organisms can be detrimental to the shelf life of both raw and processed food products, especially during the extended storage conditions. These organisms are going to have a limited ability to reduce the dyes such as methylene blue and rosagerin, and hence dye reduction tests do not correlate well with the plate counts in such cases. So we are not using this one. Then how do you enumerate the psychotrophic microorganisms? So the enumeration of these psychotrophic microorganisms can be done by preparing the proper dilutions and then plated either by the pour plate and the spread plate method on non-selective media such as plate count agar or tryptone, glucose extract agar or trypticase soya agar. and subsequently after pouring or spreading, we have to incubate at refrigeration temperature at 7 degrees centigrade for 10 days, and then we can count the number of the colonies, that is colony forming units per ml, and then you are going to find out the count of these organisms. So this is how what is the spread plate and the pour plate. Just a figure I have given you. So as you already know what is a spread plate, so you have to put the sample of 0.1 or less, and then you have to take a spreader, and with the spreader you have to evenly distribute the sample, and then you have to incubate it, and after the incubation you can see the surface colonies, and you can count it and you can enumerate it. Then coming to the pour plate, you are going to mix both the media as well as the organisms, and you are going to incubate it. Then you are going to have the surface, submerged colonies, all these things can be counted. So mainly used for anaerobic type of the microorganisms, or the microphilic organisms can be found by this kind of the pour plate method technique. Then the second type of the organism is the thermoduric. And how can we count that thermoduric organism? The thermoduric organisms are those which have an ability to survive heat at elevated or high temperatures. Example is the pasteurization. And one more thing that you have to remember is they are going to sustain, that means they will survive, but they won't grow at the high temperatures. If the organisms are going to have the ability to grow and survive at high temperatures, we call them as thermophiles. But the thermoduric organisms are able to survive at heat temperature, but they don't grow. So that's the difference between the thermoduric and the thermophile. The best example is the pasteurization conditions, where these organisms will survive but they don't grow, are going to be called as thermoduric. And the examples are Orthobacter, Bacillus, Clostridium, Enterococcus, Micrococcus, and Lactococcus are going to be the best thermoduric organisms. And these organisms are able to spoil the heat treated and the canned products by producing the acid production, proteolysis, and lipolysis. And the thermoduric organisms are going to have a special interest in dairy products. That means where they are known to survive the pasteurization. And here also we are going to have the limited ability to reduce the dye. That means these organisms have limited ability to reduce dye, such as methylene blue and visargine, and hence dye reduction tests do not correlate well with the plate counts in such cases. Then how do you enumerate? That means how do you count or grow these thermoduric organisms? So for enumeration of this thermoduric bacteria, we are supposed to take the 5 ml of volume of liquid sample in original or of decimal dilution in aseptically, and then it is going to be transferred to a sterile test tube, and a rack of such test tubes is placed in a water bath and maintained at a temperature of 62.8 degrees. So after a period of 30 minutes, tubes are immediately cooled in an ice water bath. Now 1 ml of this sample can be analyzed by a plate count method using plate count agar and incubation at 37 degrees centigrade for 72 hours. So after that you can see the growth of the organisms, and you can find how many of them are there, and the one which was grown are going to be nothing but thermoduric bacteria. The lipolytic count. So the fat-rich food products are liable to be spoiled by lipolytic microorganisms. The non-lipolytic organisms commonly encountered in foods include bacteria, where Pseudomonas, Achromobacter, and Staphylococcus. Then you are going to have the molds, which is going to be of Rhizopus, Zeotrichum, Aspergillus, and Penicillium. Then the yeast species are going to be Candida, Rhodiola, Torula, and Hansula. So the fat-rich products can be liable to be spoiled, that means it can be spoiled by non-microbial origin also. That means these microorganisms are not only responsible of lipolytic activity, there are some other factors which are influencing the this lipolytic activity. So those are all being classified as non-microbial origin of lipolysis of the concerned fat-rich products. Then how can we enumerate this lipolytic count? So the lipolytic organisms can be enumerated by counting on a base layer medium which comprises of a fat 50%, that is 50 grams, then Victoria Blue of 1:1500 solution of 200 ml solution, then agar of 15 grams is used. Now, the fat may consist of a tributylene, corn oil, soybean oil, cooking oil. Out of these, the tributylene is the most widely used substrate, and hence commercially available tributylene agar is commonly used for lipolytic count. So once you have prepared that one, the plates of the lipolytic medium are streaked with a dilution of sample and incubated at 20 to 25 degrees for 3 to 4 days. The presence of the clear zone of hydrolysis around the colony is taken as a positive indication, and inferences are drawn accordingly. And the one which is not having the clear zone are going to be taken as a negative. So here in the pictures you can see the lipolysis is going to be seen as a hydrolysis of the surrounding medium. So this is considered as a positive, and these are going to be considered as a negative. Then moving to the proteolytic count. So the proteolytic activity of microorganisms can lead to a number of undesirable flavor defects in milk, meat, poultry, and the seafoods. The best examples of proteolytic microorganisms are Bacillus, Clostridium, Pseudomonas, and Proteus. So for the growth of these organisms, the milk agar medium is used to grow and know their proteolytic activity of the concerned proteolytic microorganisms. How do we enumerate the proteolytic organisms? So for enumeration of proteolytic bacteria or the organism, especially from the dairy products, milk agar medium is commonly used. Then the hydrolysis of casein manifests into development of a clear zone around the colony. So here you can see the clear zone of this protein hydrolysis. The sample, that means whatever the sample you are going to take, are streaked on this medium, that is milk agar medium, and incubated at 21 degrees centigrade for 72 hours. And other than this milk agar medium, we can also have the gelatin agar medium with a double layer plating technique, can also be used for this purpose. And the limitation of this method is, that is gelatin agar medium, is it should be needed to flood the medium with protein precipitant to ensure that the clear zones are formed as a result of proteolysis are not due to the acid production from carbohydrates. So that is the limitation of this gelatin agar medium. So that's how you can enumerate the protein lysis or the hydrolysis of these proteolytic organisms. Then moving to the pectinolytic count. So pectinolytic enzymes are capable of hydrolyzing the pectic substances like pectin isomerase, pectin methoxylase, pectin methylesterase, and pectate lyase. The best examples of these pectinolytic organisms are Achromobacter, Aeromonas, Orthobacter, Agromobacter, Bacillus, Clostridium, Enterobacter, Flavobacterium, Pseudomonas, Xanthomonas, yeast, molds, protozoa, and nematodes. So many are there which are acting as a pectinolytic microorganisms. Then how can we enumerate? So medium used for the detection of pectate lyase is going to be of what we call as polygalactorinase and fluorescent pectinolytic pseudomonas are: number 1, mineral pectin 7, which we simply call as MP7, mineral pectin 7 medium, which we simply call as MP5, and the fluorescent and the pectinolytic agar, which we call it as simply FPA. Okay, fluorescent and pectinolytic agar. So first of all, you have to take the homogeneous suspension followed by appropriate dilutions of the food sample, and a volume of not more than 0.25 ml is placed and spread on the surface of the pre-poured plates. So we can take any media, that is MP5, MP7 or FPA. So you have to place the 0.25 ml prior, which is already diluted, and you have to spread on the surface of pre-poured plates, and you have to carry the incubation after the pouring of about 48 hours at 30 degrees centigrade. Now, the colonies which affect the depression in the polypeptide gel medium are found to be enriched by a clear zone after flooding the MP7 or MP5 or FAP plates. A pectin precipitant are considered as pectinolytic colonies. So, these are going to be considered as the pectinolytic colonies and they are counted. So, then moving to the halophilic count. So, halophiles are those organisms which need certain minimal salt concentrations for their growth. And these halophiles or halophilic microorganisms are going to be classified into three types depending upon their salt concentration. That means NaCl concentration. So if they are going to be of having the 2 to 5% of the salt concentration, they are going to be called as slight halophiles. The examples are Acinetobacter, Flavobacter, and Pseudomonas. Then if it is the salt concentration is 5 to 20%, then we are going to consider them as a moderate halophiles. The examples are Basilaceae and Micrococcaceae. And the salt concentration is very high of about 20 to 30%. We call them as extreme halophiles. The best examples are Halobacterium and Halococcus. So how can we enumerate these microorganisms in the food samples? So first of all, we'll see the slight halophiles, then we will move to the moderate and the high. So the first one, salt halophiles, the best examples already I told you, the Acinetobacter, Flavobacterium and the Pseudomonas. So these are going to be of a main spoilage of marine products and the shellfish. That means these organisms, the slight halophiles, are responsible for spoiling the marine products and the shellfish. For enumeration of these microorganisms, the skins are removed from fish. That means the samples are going to be taken from the skins of the fish, and the 6 samples are collected, 3 each from the dorsal and ventral side. So 3 from the dorsal side and 3 from the ventral side we are supposed to take. And 10 grams of the skin is going to be added to 90 ml of the phosphate buffer. So out of the 10 grams is going to be added to the 90 ml of the phosphate buffer. And now the sample is vigorously mixed and subsequent dilutions are prepared in the phosphate buffer only. So you have to prepare the serial dilution. A volume of 0.1 ml aliquots is dispensed on the trypticase soya agar plus, which consists of 3% NaCl, and evenly spread on the plate using spread plate technique. So again you have to go with the spread plate. And the plates are incubated at 7 degrees centigrade for 10 days and the results are reported as colony forming units per gram. For light salted meat and vegetables, dilutions are prepared by blending the 50 gram sample with that is what we call as 450 ml diluent with the added salt concentration. So for the fish, we have taken the phosphate buffer. For the meat and the vegetables, we are going to have the diluent with 450 ml of with added salt concentration equivalent to the salt concentration of food sample. And the rest of the procedure is the same. That means you will take the 0.1 ml of the dilution and you will plate it and incubate it for 7 days at 7 degrees centigrade for 10 days and you will observe for the colony formation. So the process is the same. Then how do we enumerate the moderate halophiles? That means the halophiles which grow at the 5 to 20% of salt concentration. So here 1:10 dilution sample is prepared by mixing the 50 grams of food in 450 ml of sterile buffer. So previously we have taken the phosphate buffer. Here we are taking the sterile buffer with the added NaCl equivalent to the salt concentration prevalent in the food sample. Now from this dilution, we are going to take the 0.1 ml and it is dispensed on the trypticase soya agar, which consists of 3% NaCl, and it is going to be evenly spread on plate using the spread plate technique. And now here the plates are incubated again at 7 degrees centigrade for 10 days and the results are reported as colony forming units per gram. Then the enumeration of extreme halophiles, that means the one which grow at the concentration of 20 to 30% of salt then those are going to be called extremophiles. Examples are halobacterium and halococcus. And these are going to be usually found in aquatic environments of unusually high concentration, so marine concentrations. And in solar evaporated sea salts, we can find these bacteria, that is halobacterium and the halophiles. So for isolation of these extreme halophiles, the surface lime from salted fish is transferred to a halophilic agar plate using a cotton or an alginate swab. That means you will take a swab or a cotton and you will rub on the surface of it, and that is going to be taken as a sample. And for enumeration, 1:10 dilution is prepared by mixing 50 grams of sample with 50 ml of halophilic agar and incubated at 3 to 35 degrees centigrade for 5 to 12 days in a humid incubator. And the results, as we know, we are going to take it as a colony count, that is colony forming units per ml or per gram. So this is how you're going to have the halophilic count. Then moving to the osmophilic count, yeast capable of growing in high concentration of sugar happen to be the common osmophiles found in our food. And these osmophiles are the one responsible for the spoilage of our honey, chocolate candies, jams, molasses, corn syrup, fruit juices, and the similar products. That means if we are having the spoilage of these products, then we can assess that it is going to be because of the yeast organisms responsible of spoilaging these products. Then how can we enumerate them? So the enumeration of osmophiles are going to be done by appropriate dilutions prepared in phosphate buffered solution, water as a diluent. Then they are going to be plated in agar. That means the sample of 0.1 is plated on the agar, like potato dextrose agar, malt yeast extract agar with 40% sucrose. As it is a yeast, we have to take the potato dextrose agar and then incubated at 30 degrees centigrade for 4 to 5 days. Then we can observe the yeast growth. There you can enumerate and find the colony forming units, and we can find the osmophiles. Then acidophilic count. The acid producing bacteria are found in habitats like soil, raw agriculture products, and processed foods. This lactic acid bacteria are going to be the best example of acid production from sugars present in the substratum, mainly useful in food industry. The lactic acid bacteria are going to be of two types, that is homofermentative and heterofermentative, where homofermentative means the final product is only one, that is lactic acid, whereas in the heterofermentative, along with the lactic acid, it may produce the ethanol or acetic acid like that. So those are going to be called as heterofermentative type of bacteria, lactic acid bacteria. And these lactic acid bacteria happen to be the major estophiles in our foods. So other than these lactic acid bacteria, certain bacteria are also responsible for producing the acid, producing like bacillus, clostridium. These are the best examples of spore forming bacteria. And the enteric bacteria like E. coli, Enterobacter, Salmonella, and Shigella. Then how can we enumerate these acidophilic bacteria? So for detection of acid producing bacteria, there should be a determination of titrable acidity. That means acidity expressed as lactic or acetic acid. And indicator microbiological medium which contains a pH indicator, best example is the bromocrystal purple or required. Now, the lactic agar or M17 agar or MRS agar are used for selective growth and enumeration of these lactic acid bacteria, mainly streptococci and lactic acid or lactobacilli. Now you have to, that means using the pour plate method, these medias, that is lactic agar or M17 agar or MRS agar, can be used for direct enumeration of lactic acid bacteria. Now, once their identity can be further confined by staining and biochemical tests like gram staining, catalase test, etc. So, the measurement of titrability of the acidity can be used as an indirect method of estimation of this acid production. So this is all about the enumeration of acidophilic count. So we finish the enumeration of different types of the spoilage microorganisms in the food. Okay, so like acidophilic, lipolytic, psychotrophic. So all these types we have discussed in detail. And in the next part, we are going to detect. In this video, we had gone with the enumeration. In the next video, we'll go with the detection of pathogenic microorganisms in the food in detail. Thank you.